All right, before we start today, a few announcements. So another positive piece in this class, and I don't, let me turn this down. Okay, I don't have a seating chart, obviously, to contact trace, but the student has, the person she was sitting right next to knows, and she said she was sitting on the right side, third or fourth row. So if you're near there, if you were here on Tuesday, just, you know the drill, be extra vigilant, and just don't ignore any symptoms that you have, get tested. You don't need to quarantine if you're vaccinated, but you know, it is what it is. So, right, we have masks on, but you know, Delta doesn't necessarily care about that. So just being as upfront with you as I can be. All right, so this area over here. Drop day is a week from tomorrow. So October 29th, that's a drop deadline. So just want to point that out. If you want my help in running the numbers to see kind of where you're at, send me an email and I can kind of help you think through that a little bit. If you're thinking about dropping lecture and you're doing well in lab, talk to your lab professor. You just need to get a course override to stay in lab if you want to drop lecture. Because if you drop lecture, they'll automatically drop you from lab because lecture is considered prerequisite for lab. So, but we approve that all the time. Just kind of keep that in the back of your mind there. Okay. And obviously no class on Tuesday. We will be here next Thursday, but fall break on Tuesday. I'm sure you're all aware of that. Okay, and I just wanted to, I'm looking ahead. We have an exam actually 2 weeks from today. So not right when you get back from fall break, but the week after that. I'm kind of looking ahead and thinking about what's going to be covered on that exam. And I'm a little worried I'm going to be squeezed for time. So I'll try to make the decision maybe this weekend, whether I need to like post a little extra material. I'd rather do that within the next, 3-4 days, rather than, obviously I don't want to do that right before the exam. So I know that's not like a popular thing to say, oh, here, we're not going to get to this in lecture, listen to this on your own. But we are losing a whole lecture from fall break, which I'm kind of used to having. So again, I know that's not popular. It would be a short video. If there is one, we'll see where we're at. We'll get through with what we can. All right. I did want to take just a few minutes though, and now that I think everybody that's going to take the exam has taken it. I wanted to talk through a couple of the questions that were commonly missed from the last exam. Had a good number of you come and look at your exams in my office, so you can continue to do that. These 3 questions kind of stood out to me. I knew that the first 2 would be difficult, but the third one I didn't really expect it to be, and it was. All right, so referring to the diagram here, which strand is the coding strand? And I said that this is transcription. If that should have been obvious because only one strand of DNA was being copied. But assuming that this is the strand that's being copied, that's the template strand. And so this strand up here, A, would be the coding strand. It's going to be the same code as this RNA that's being made. Okay, because this RNA is going to be complementary to this strand, just like this strand is complementary to this strand. All right, I know it's a little bit confusing, but the coding strand would be A, and which letter denotes the 3' end of the DNA? Well, you know that the new RNA, it's always being added on to the 3' end. So this is 3' end of RNA. So this is going to be a 5' end of DNA, and those DNA strands are anti-parallel, opposite directions. So this is a 3' end. So the answer again there is A. All right, referring to the same diagram, what enzyme? What is the major enzyme of transcription? That's RNA polymerase. All right, and I guess maybe these two enzymes kind of sound the same, but only about a third of you got this question right, and we only had like 50-50 for these two. So when there's only two choices, I was hoping to get a little bit better than that. But okay, so those three steps tripped some people up. This question about coenzymes, I wasn't expecting this one to be difficult either, because it was just kind of a definition. What's a coenzyme? I made up part A. So I guess that kind of sounded right, because coenzyme sounds like it's together with something else. But I made that up. Coenzymes are organic cofactors. So a cofactor, remember, is something that's not a protein that is necessary for the enzyme to function. A cofactor is typically like a heavy metal, like iron maybe, or copper. If that cofactor is organic, then we call it a coenzyme. If you're in lab, and we're going to talk about this later today, but if you're in lab, you might have talked about it in lab, folic acid is actually a coenzyme. Folic acid has to be there for an enzyme to function in the production of nucleic acids. So that's just an example. We'll talk more about that today. And one last one that I wanted to bring up, lactic acid bacteria, a group of bacteria that live in the human body, and they lack cytochromes. And a lot of people said to me, I had no idea what a cytochrome was. So cytochromes are actually present in the electron transport chain. So the correct answer there was respiration. When did we talk about that? This slide should look a little bit familiar. I said that you don't need to memorize the order of these electron carriers, but you should recognize the kinds of molecules that they are. All right, and I thought cytochromes would be the most recognizable of those. But again, that must have been something that was kind of glossed over. So that's where I got that question from. All right, so again, come and look over your exam anytime. If my office hours don't work, then we'll find another time to stop in, ideally before the next exam. All right, questions, comments? All right, so last time we started antibiotics and talked a lot about kind of a little bit about the history, you know, kind of where they come from. Different things we have to think about when we administer antibiotics, like how safe they are, allergies. We did talk already, we started talking about the beta-lactam antibiotics, and we mentioned that, hey, allergies are pretty prevalent with penicillin and beta-lactams. So that last slide that we ended with, it was somebody's back or chest, I forget, that had a bad case of hives. So that's a common side effect, maybe a little rash or hives, can be severe. Potentially though, the next time that person has a reaction to penicillin, it could be a lot worse. It could, you know, person could go into shock. And 300 to 400 people in the US die of hypersensitivity reactions to penicillin every year. So if your provider, your doctor knows that you had a reaction at one time, they will not prescribe a beta-lactam antibiotic again. And what I meant to say, and I finished up before I said it last time, was that if you are allergic to penicillin, then typically you are allergic to all the beta-lactams. Okay, so that's methicillin, that's amoxicillin, what we're going to talk about now. It's not just penicillin, and there are several more that we'll talk about as well. So usually, again, if you're allergic to one, all those compounds are related. They all have that beta-lactam ring right here. Remember, this is a beta-lactam ring. That's what enables them to fit into that active site of that enzyme transpeptidase. Probably going to have a reaction to all of those. All right. So the penicillin and the methicillin that we talked about last time, those are pretty narrow spectrum antibiotics, meaning they only target some gram positives. Amoxicillin, we came out with amoxicillin in the early 1970s, and it was one of the early beta-lactams that had a little bit broader activity. So it doesn't just work against gram positive, it works against some gram negatives as well. And probably most of you have had amoxicillin at some point in your life. It's that they put bubblegum flavor in it, so it's bright pink. It tastes good. So, you know, really ideal for some of those common childhood infections or infections that we get as adults too. So things like strep throat and ear infections. Okay, those, you know, just kind of the common things that we just kind of come down with every once in a while. Like I mentioned last time, my daughter, who's 24, came down with an ear infection last weekend, and they prescribed amoxicillin, and I thought, oh, she's going to get that pink liquid. Well, when you're an adult, they give it to you in pill form, but those pills are enormous. I was really surprised. They're bright pink, but she's got to swallow this enormous pill. It seems to be doing the trick, though. She's doing good. So amoxicillin, you know, again, it's very commonly used. It's acid stable, meaning we can take it orally, we can swallow it, and it won't get broken down with our stomach acid. The dosage is not very common because it absorbs pretty well from the large intestine. So she's taking it twice a day. That's not too bad. The one thing that we worry about, like amoxicillin's been in use, like I said, since the early 70s. So that's a pretty long time. We are seeing some resistance. So in an effort to prolong the use of amoxicillin, it's very often packaged with clavulanic acid. And what that is, it's a beta-lactamase inhibitor. All right, so we talked about that last time, what beta-lactamase is. That's an enzyme that bacteria acquire the ability to make, and that enzyme cleaves the beta-lactam ring right there, opens that up, and it basically destroys the activity of the beta-lactam drug. Okay, so if the bacteria can make beta-lactamase, then they can survive in the presence of the drug. All right, so we don't want bacteria to be able to make that. If we, again, we package the amoxicillin with this clavulanic acid, that's kind of supposed to block the action or protect that beta-lactam ring a little bit. And when we package those together, they're in the same pill. It's all mixed in there. You don't take two separate pills. We call the drug augmentin. Okay, if you augment something, that means you add to it to make it better, right? So we've added clavulanic acid to amoxicillin to give it hopefully a longer lifespan and hopefully slow resistance of microorganisms to the drug. So pretty often you see that brand name augmentin. Okay. Let's see. Other beta-lactams. I think this is just the one other one that I wanted to talk about. The cephalosporins are in very wide use. You can see they have that beta-lactam ring. They have two side chains now that are variable. So we have a lot of different cephalosporin drugs. They all start with this prefix cepha, and either CEPH or CEF. So like cephalexin, ceftriaxone, you see that prefix, and that's a hint that it's a cephalosporin. The name came from the organism, Acremonium cephalosporium, that naturally secretes it. It is confusing getting back to the names of drugs. We often, the company that manufactures it likes to put its own trade name on drugs. So for instance, cephalexin, the trade name is Keflex. All right, so that's the same thing. Just, I don't know why they like to put their own name on there. So you may have heard of Keflex, pretty widely used. Okay, so it's a beta-lactam. It binds those transpeptidases, leading to cell wall lysis by osmotic pressure. And when we first came out with these, we thought the structure was going to keep them in use longer. As far as I know, there hasn't been a lot of resistance to cephalosporins. We have actually many different generations. There's, I think, 5 different generations of cephalosporins. So the first generation were the ones that we made a long time ago. We keep tweaking them a little bit. Again, they're semi-synthetic, I believe. So we can, kind of tweak one side chain or another to give the drug a little different properties. So a lot of the properties of the drug, it depends on what generation that you're talking about. The earlier generations were very safe. They were fairly narrow spectrum. So low generations only targeted gram positives, and they were pretty safe. The kind of newer ones that we've come out with have been more broader spectrum, meaning killing some gram negatives as well as gram positives, but then had a little bit more toxicity concerns with them. So the later generations, we have to dose out very carefully and usually are only given by IV. But some of the higher generations, I think at both the 4th and 5th generation, they have this distinct advantage of being able to cross the blood-brain barrier. There are only a few pathogens that can do that, but we need to have drugs that can follow those pathogens there. So again, the later generation cephalosporins we can use to treat things like meningitis. And also the 5th generation is kind of different enough that seems to work pretty well against that methicillin resistant Staph aureus. And oddly enough, even though I just told you that if you're allergic to one beta-lactam, you're allergic to all of them, apparently the later generation cephalosporins, some people that have a penicillin allergy are able to tolerate those later generation cephalosporins. To me, that's not good enough. Oh, here, you might be able to tolerate this. Like, that wouldn't be good enough. They would probably do some kind of test to see if you had a reaction to it, unless your life was in danger and they just had to give it to you. But, all right, so that's the beta-lactams. There's a couple other drugs I want to talk about that also target the cell wall. They're not beta-lactams, so they work in a little slightly different mechanism. But again, good selective toxicity, because remember, we don't have cell walls, so they're targeting a structure that the bacteria have that we don't. Vancomycin is this really large structure here. All right, not a beta-lactam. How does it work? It's kind of more of a physical obstruction. So it binds on those same peptide bridges within that peptidoglycan structure and just kind of blocks their formation again when you have the growing cell wall. Only works on gram positives, so that's considered pretty narrow spectrum. We used to use vancomycin. I think it still pretty much is considered kind of a drug of last resort. So you try everything else and it doesn't work. All right, let's try the vancomycin. It has less toxicity issues than what it used to. We got better at purifying it, but still kind of used like, okay, nothing else works, we'll try the vancomycin. It does often do well against MRSA. The other thing we use it for is to treat C. diff, Clostridium difficile. So that really nasty diarrhea sometimes ulcerates the large intestinal wall. This drug is pretty poorly absorbed, so usually it's either injected or given by IV. But in the case, the exception to that is when you're trying to treat C. diff, patients have to swallow it to get it to the large intestine where it kind of needs to be to treat the organism. All right, so again, usually a drug of last resort, but in recent years, we have found strains of bacteria that are even resistant to vancomycin. So we have this Enterococcus. If you're in lab, we've used that just a couple of times. Enterococcus is similar, very similar to Streptococcus. In fact, we used to call it Streptococcus. We decided that these, few Streptococcus weren't typically causing disease, so we put them in their own genera. Well, it turns out that they can cause disease if they're in kind of the right part of the body where they don't really belong. But now we have these strains that are even resistant to vancomycin. So this, if you're in the hospital, typically with some other issues, and if you maybe get some VRE, vancomycin resistant enterococci in your bloodstream, that's a medical emergency. Like, they don't have good options to treat that organism. So that's one of those super bugs that sometimes you hear about on the media. Bacitracin, another drug that targets the cell wall. This actually prevents those sugar subunits, NAM and NAD. They're synthesized inside of the cell, and then they have to move across the cell membrane to get out to the cell wall. Bacitracin kind of stops up that channel that they move through. Okay, so it keeps the NAM and NAG from passing across the cell membrane to the cell wall. Really narrow spectrum, once again, only gram-positive. We don't actually take this drug orally because it has such high toxicity to the kidneys. Okay, so we only use it topically. So it's used for skin infections. Major use as part of triple antibiotic cream. Okay, so now it's recommended, if you get a cut or you scrape your knee, to put some triple antibiotic cream on there, rather than using something like hydrogen peroxide would work as well. But this is one of the antibiotics that you'll find in triple antibiotic cream. All right, we'll get to the two other ones shortly. All right, one more cell wall inhibitor to mention, and it's one that's kind of fun to say, Isoniazid. Isoniazid. This is a totally synthetic drug. So most of the antibiotics in use are either totally biosynthesized by fungi or bacteria, or semi-synthetic, meaning we kind of mess with the side chains a little bit, but this is one that's totally synthetic. Remember, you don't, that's one of the details that I don't need for you to memorize. So I'm talking about, how things are administered, whether they're acid labile, just to kind of get you used to thinking in those terms. You don't have to have each drug memorized, how you made it and where you put it. Okay, so I want you to focus on the mechanisms of action. In this case, it's preventing the synthesis of mycolic acid. Focus on maybe uses or nasty side effects. Okay, so prevents the synthesis of mycolic acid. That, knowing that about this drug tells you that it's going to be very narrow spectrum. Okay, the only organisms that have mycolic acid in their cell walls, okay, those are going to be useful against our acid-fast organisms. And that the main genera is the Mycobacterium. All right, so if you treat, Staphylococcus with Isoniazid, the Staphylococcus is going to be just intrinsically resistant to that because you're targeting something that the staph doesn't do. All right, but works against Mycobacterium, so it's going to be used to treat things like tuberculosis and leprosy. Those are the two main. There are lots of other Mycobacterium infections, but those are kind of two of the most famous ones. Yes, question. So how do we use antibiotics against gram negatives if they release endotoxin? We're going to get to that when we talk a little bit more about toxins after break. Very carefully is the answer to that. So if you have a gram-negative infection, a really bad one, oftentimes we try to let it go untreated if we think the immune system is going to be able to take care of it itself, or we give enough anti-inflammatories to kind of counteract the effect of endotoxins. But yes, we will talk more about that. That's a good point to bring up. All right, so pausing for a minute. I know I have just run through a lot of 7 different drugs there. I think a good way to kind of think about all these drugs, rather than doing something like flashcards, is to organize our information in terms of a concept map. All right, so I've started one for you here. If we take, antibiotics, and then we think about our 5, we've got 5 different mechanisms that we're exploring. Within the cell wall inhibitors, we've got 4 that we're going to talk about that are beta-lactams. We had 3 other ones that we just mentioned. All right, so and then as we go along, you can fill in, some of our other drugs that we're going to talk about here. So I think you could write other things in here, like, this is the one that blah, 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 does this or that. You can fill in a little other information there, but this is good practice actually to write this out multiple times. All right, so I'll come back to this when it's all filled in, but you should be able to fill this in yourself, kind of go back in your notes and fill in as we talk about them. All right, so moving on to the next really big class of antibiotics, protein synthesis inhibitors. Okay, so remember where protein synthesis takes place. That's the ribosome of our cells, the ribosome of bacterial cells. Bacterial ribosomes, remember, are 70S, ours are 80S. So similar structures, slightly different structure, different enough that we have antibiotics that will adhere to bacterial ribosomes and not so much to ours. Okay, so they adhere to the bacterial ribosomes with a greater affinity. Okay, so in general, the drugs that we have that are protein synthesis inhibitors, most of them are quite broad spectrum. And remember that you might think about, that's a good thing, it just kills everything. But remember, we have the chance of maybe having some side effects from that, like it working on our normal microbiota and causing the variability of our normal microbiota to get a little bit out of whack, so that we might get, C. diff or a yeast infection or something like that. Okay, so it has its advantages and disadvantages to be broad spectrum. And that bacteria that we said kind of acts like a fungi, that genus Streptomyces actually produces a whole bunch of the antibiotics that are considered protein synthesis inhibitors. All right, so our first class that we're going to talk about are the aminoglycosides. Okay, so what can we say about this group of drugs? They are all going to bind to the shape of the small subunit of the ribosome. Okay, and I'm going to show you some examples of how some of these protein synthesis inhibitors work, but if you just know like that they bind to the ribosome, then that's probably going to be close enough. Like I said, they're broad spectrum. They don't absorb very well when we take them orally through the small intestine, and they are impacted by our stomach acidity. Okay, so that means they have to be injected. Fairly high toxicity with aminoglycosides. And I found this nice little mnemonic over here that reminds us of that. So you see an ear boxing with a kidney. And you can't see it very well, but on those boxing gloves is written the names of some aminoglycosides, like this one is gentamicin. All right, so our ears and our kidneys can kind of take a little bit of a beating. Those are the side effects that are associated with aminoglycosides. And some examples here, kanamycin, gentamicin, streptomycin, they're ending in myosin. That's typically, we know the ends in myosin, and that's probably an aminoglycoside. They don't always spell it the same, and that's a nod to a different organism actually secretes gentamicin than what secretes streptomycin and neomycin. So there is a reason that that one is spelled differently. In case you were wondering, that's why. So we're going to talk a little bit more about streptomycin. I wanted to mention neomycin because it's our second antibiotic in triple antibiotic cream. Okay, so we've got the bacitracin that works on bacterial cell walls. Now we have a protein synthesis inhibitor. And then the third antibiotic, like you can guess, is a different mechanism of action. So it's really trying to hit bacteria from lots of different angles. Okay, so streptomycin, here is a structure of streptomycin. So remember, this is an example of an aminoglycoside. The structure of an aminoglycoside just means amino modified glycosidic sugar. So when you look at this structure, you see all these amino groups, amino modified glycosidic sugars. This is another way to draw sugars. These are 6 carbon sugars. Okay, so usually the name kind of makes sense when you look at the structure. Should you memorize that? No, but look at it and know, okay, well, this is not a beta-lactam. I don't see a beta-lactam ring, and I see some things that I recognize, okay? Streptomycin binds to the small subunit. So you can see your book's little picture here of how that works. So it's working at the level of translation. It's binding to that small subunit there, and it just kind of changes the shape of that subunit enough that the messenger RNA gets misread and does not make the correct protein. Okay. So again, eventually enough of that builds up and the cell's not going to be functional anymore. So like I said, broad spectrum, toxicity to ears. So it seems like an odd side effect. It actually binds to, those of you that are in anatomy will appreciate this, the vestibular portion of cranial nerve number 8. Okay, so that's where streptomycin binds. So that can cause things like ringing in the ears, tinnitus, maybe a little vertigo where you get like nauseous from the ear trouble, can cause deafness that can be transient but can be permanent. All right, so we have to be very careful about dosing streptomycin out too, right? You give too much and the patient can be deaf as a result. But streptomycin, we don't use it anymore unless a patient's life is in danger because of that ear toxicity. That's a side effect that we don't want. But streptomycin, when it came out, it was one of the earlier antibiotics that came out in the 40s. And when it came out, it was a miracle drug because it was effective against tuberculosis. So an infection that we had had no cure for thousands of years, suddenly we had something that worked against it. And Bob Dole, a lot of you don't even know who Bob Dole was. He was in the Senate for years and years. He was a child, had TB as a child, and was cured with streptomycin, and went on to live this really long, productive life. So, all right, so we don't use it for TB anymore, again, unless the patient's life is in danger. But it also was the first drug that was effective against bubonic plague. All right, bubonic plague is caused by a gram-negative organism. So this was really the first drug that worked against gram-negatives. Tularemia is less common, but that's a drug that, that's an infection that's carried by the fleas that are on rabbits. It can cause, and that can be a really life-threatening infection as well. So again, miracle drug. Side effects are such that we use other things whenever possible now. Other protein synthesis inhibitors, tetracyclines. Again, look at the structure here, 4 rings. Okay, makes sense. This also, tetracyclines are, again, another huge class of antibiotics that target the 30S subunit. And let me just skip ahead to your book's graphic as to how that works. But it binds here to the 30S subunit and makes it so new tRNAs can't dock at that A site. So it's going to stop protein synthesis. Broad spectrum drug, widespread use because it's so cheap. And that's one of the reasons that it's had such uses in agriculture. We found out, we used to give antibiotics to farm animals, right, when they got sick. We figured out that if we put antibiotics in their feed, that would keep them from getting sick. And it also had the side effect of making farm animals grow faster. So great, increase the profit margin of farmers. Unfortunately, we're worried that that's one of the reasons that we get such antibiotic resistance today. So we're trying to cut back on the use of antibiotics in animal feed. Again, it's difficult to do because farmers make more money when they can use it. So it's kind of this huge problem. We are successful in the United States. I know California has outlawed putting antibiotics in animal feed. It's worldwide that we don't have as much control over. It can be taken orally. There are some nasty side effects, right? So you can't avoid that picture there. The major side effect that we think about is that it impacts developing bones and stains developing teeth. So this person probably had tetracycline as a child, and now their teeth are really discolored as a result. So we don't give tetracycline to children. Again, unless their life is in danger and nothing else is working. The other side effect that's kind of common is phototoxicity. So that means if, say, you're on an acne medication that has tetracycline in it, you have to be really careful when you're outside in the sun. You're going to get sunburn much easier. So anytime you're outside, you got to lather on the sunscreen. I was, just before we came into class, I was looking up side effects of tetracyclines a little bit more carefully because my older daughter called this week, and she has this bug bite on her shoulder that was looking kind of suspicious. It was growing and growing. So earlier this week, she went to the doctor, and he said, "I'm a little worried this might be Lyme disease. So we're gonna put you, from a tick bite. So we're gonna put you on doxycycline, which is tetracycline. It's a tetracycline." And so, okay, good. You want to treat that, get that out ahead of that. You don't want to realize like weeks later that yes, it was Lyme disease, because then there's less that you can do to deal with it. And there's some, neurological things that we don't want to deal with. So yes, treat it, hopefully get rid of it. But I was looking up the side effects, and the first two are nausea and diarrhea. She's flying back to Grand Rapids this weekend to run the 5/3 race, the 25K. So here, I'm keeping my fingers crossed that she's not really feeling lousy to go run 15 miles. So I'll keep you posted, and we'll keep our fingers crossed. And yes, we want to get rid of any possible Lyme disease, but hopefully we won't have to deal with those side effects. All right, so uses for kind of specific infections, not something that you give out kind of every day for any little thing, basically, at least in humans. All right, and one more protein inhibitor, a class of drugs called the macrolides. So I don't know much about the chemistry here, but this is considered a macrolide ring, and that's why they call them macrolides. These are drugs that actually target the large subunit. So our tetracyclines, our aminoglycosides, they're all targeting the small subunit. Macrolides target the large subunit. Your book talks about a lot of antibiotics that, we're going to focus on the ones that we're talking about in class. So here, macrolides binds, blocking proper messenger RNA movement through the ribosome. So again, translation can't happen, new proteins aren't made. The macrolide that I want to use as an example, the one that's most commonly prescribed is azithromycin. And you might have heard that called a Z-Pak. Very commonly prescribed medication, especially if you are allergic to the beta-lactams. Probably, I think, one of the most common, again, prescribed antibiotics. So we use it for things like ear infections and strep throat pretty commonly. Also some more serious infections like gonorrhea in combination with, I think, one of the tetracyclines. We have a lot of problem with resistance to gonorrhea, so we've kind of cycled through a lot of antibiotics. I think right now it's azithromycin and doxycycline. We'll talk more about gonorrhea later on in the semester. Fairly broad spectrum. Really nice thing about this drug, you can take it orally, and because the concentration stays pretty high in our tissues, it's pretty long acting, you only have to take it once a day. So I think it's the Z-Pak is 5 pills, you take it once a day, and you're done with it. So that's really kind of a nice thing. There are some allergies to this as well. Some GI issues, a little nausea, but much less than the macrolide that used to be prescribed, erythromycin. I remember taking that when I was a kid, and it just cramped me up so bad, like in my stomach. You just felt like you couldn't eat anything. So there is less of that with the erythromycin than kind of the earlier generation macrolide that people used to take. Now, you may have heard of azithromycin. About a year ago, they were kind of, it was in the news, hey, let's use azithromycin to treat COVID. And you should think, wait a minute, this is used on bacteria. Obviously, COVID is viral. That seems crazy. Well, it turns out that azithromycin has a little bit of, they were hoping that had a little bit of antiviral activity. They know that azithromycin actually increases some cytokines of our immune system. So they were kind of hoping that since we already knew that azithromycin was safe, that we might be able to use it for COVID to help too. But I think basically the verdict is that it didn't really do any good. So and you might have seen that in the news. Okay, so a couple of other mechanisms that we want to discuss here. There are drugs that inhibit DNA and RNA synthesis. Again, we have to target enzymes that are kind of unique to bacteria or different enough from our enzymes so that we have that selective toxicity. Rifampin is a drug that blocks RNA polymerase. So this is working at the level of transcription. Remember, eukaryotes have a different RNA polymerase to make different kinds of RNA. So we have a polymerase for ribosomal RNA, a polymerase for messenger RNA, and one for tRNA, where bacteria just have one. So apparently that enzyme is a little bit different for bacteria than it is for us, and the rifampin binds to the bacterial kind. Semi-synthetic, easily absorbed. The one kind of shocking side effect that we see with rifampin is that it turns bodily fluids orange, right? So let me show you a picture of a patient's catheter bag there. So again, it's not bad. It doesn't harm anything. It's just a little bit shocking. It turns bodily fluids orange because the drug itself is very orange. So again, that's one of the drawbacks. That's just a little bit shocking. Also causes some vomiting and nausea, loss of appetite. So it has some other side effects as well. Primarily, we use, oh, questions. So it could be in use for bladder infections. I hate to comment too much on anything clinical that I haven't read about for sure, but if you know that it turns it orange, that could be the case. Yeah, good question. Another question? That's right. Your sweat and tears turn a little bit orange as well. So yeah, I know it would be a little shocking. It probably depends as maybe the dosage to some extent too, but you know. All right, so besides, perhaps bladder infections, I would have to look into that. The other major use of rifampin is for tuberculosis and leprosy. Okay, so typically you use, anytime you're trying to treat tuberculosis, you don't just use one drug, because we have terrible problems with resistance to treat tuberculosis. You use it in combination, usually with 4 different drugs. So rifampin is one way, and we mentioned isoniazid. Isoniazid and rifampin are kind of the two that are always tried first for tuberculosis. And the side effects here, vomiting, nausea, aside from the orange bodily fluids, those last. And when you're taking drugs, when you're trying to cure tuberculosis, you usually have to be on your drug regimen for at least 6 months, not 9 months, if not longer than that. So to experience, nausea for 6 or 9 months is, that's one of the issues that we have with treating TB. They quit taking their drugs, obviously, because the drugs are making them feel lousy. So that's why we still have issues with TB, and we haven't been able to eradicate it. All right, and some drugs that interfere with DNA replication, quinolones and fluoroquinolones, they target DNA gyrase. So if you remember, see if I can draw this a little bit. So here's, this is supposed to be a replication fork right here, and here's the DNA upstream. DNA gyrase works upstream of the replication fork. Remember, as you're opening or melting or unzipping those 2 strands, the upstream gets DNA gets kind of curled up. A lot of mechanical stress. Eventually, you can't pull those 2 strands apart anymore. So that what the DNA gyrase does is cut those strands, unwind them, and reseals them again. So without the action of that DNA gyrase, eventually replication can't continue anymore because of that kind of supercoiling that takes place upstream. And bacterial DNA gyrase is apparently a little bit different than the enzymes that we have that kind of do the same thing. The spectrum here, it kind of depends on the generation. Our early drugs, which we call the quinolones, they only worked on gram negatives. And I bring that up because next week in lab, if you're in lab, you're going to be using nalidixic acid when you're only wanting to get gram positives to grow. So you kill off the gram negatives, selective media with the nalidixic acid. The later generation quinolones, we call fluoroquinolones because we've kind of monkeyed with the structure a little bit and added a fluorine right here, where I think there was a hydrogen. I'm just guessing. Okay, so with the fluoroquinolones, just that simple adding that fluorine gives the drug a little bit better penetration, a little bit broader spectrum, just kind of made it work better and have a little bit less side effects. Okay, so we pretty much don't use quinolones anymore to treat humans. We pretty much use fluoroquinolones. The fluoroquinolones all end in oxycin. So something like ciprofloxacin, you see it ends in that oxycin, then you know it's a fluoroquinolone. So the fluoroquinolones are absorbed pretty well and they're stable in acid. We do reserve these for more serious infections. So maybe a UTI that's not responding to other drugs, sexually transmitted disease, serious respiratory infections, anthrax. If somebody is known to be exposed to anthrax spores, they give you ciprofloxacin just to make sure that you don't get anthrax. I was reading a little bit more about side effects here, and it has some really kind of bizarre side effects. One being tendon disruption. So we don't really understand why that happens, but you're more likely to tear your Achilles tendon if you're on fluoroquinolines. And we don't understand if it decreases blood flow to the joints or maybe impedes collagen formation. We don't really understand why that is, but that's kind of an unusual side effect, especially if you're an older adult. We don't give fluoroquinolines to kids anymore because we've seen some kind of serious neurological side effects like seizures or maybe nerve damage in children. Aortic rupture, that's obviously a really bad side effect. I was reading, as of about 5 years ago, the FDA put its most serious warning on fluoroquinolines, and basically we only use these if nothing else is working. One epidemiologist said, overuse of these drugs is like trying to kill a fly with an automatic weapon. All right, so again, we only use them when conditions are pretty dire, or some infection is pretty dire. All right, and I saw this mnemonic online. I thought I'd share it with you. The queen stops the gyrating dancers. So queen for quinolone, stopping DNA gyrase. Okay, I don't know if that mnemonic helps at all. Remember the mechanism of action there, then I just thought I'd share that. All right, so we just have a couple more. I know it's like filling your head with all these drugs. It kind of seems like a lot. Cell membrane inhibitors. Not a lot of these, because our cell membranes are very similar to bacterial cell membranes. So we have such high toxicity issues here that they're not in major use. Colistin is one, again, that you're going to be using in lab. So I just put an example, I threw that one up there. The way polymyxins work, they insert in the membrane. So you can kind of see, this is kind of the tail of the structure that inserts right into the membrane. And remember, anytime membranes are disrupted, that's really bad for the cell. They can't generate the ATP that they need during respiration. Things are passing in and out of the cell that aren't supposed to. So that's generally pretty bad. They're more active against gram negatives because remember, they have that outer membrane that's going to be disrupted as well. Like I mentioned, huge toxicity issues, especially in terms of kidney toxicity. So kidney damage there is the main reason that we only are going to use topically. Okay, we don't ingest polymyxins, but this is the third antibiotic in triple antibiotic ointment. So bacitracin, neomycin, and now we have a drug that works on cell membranes as well. So again, hitting bacteria from 3 different mechanisms hopefully will do the job and keep your wound clean, keep it from getting infected. All right. Lastly, there is at least one metabolic pathway that bacteria have that we don't. Okay, so we can capitalize on that by using sulfa drugs. All right. Okay, there we go. All right, so sulfa drugs came into production about 10 years or so before penicillin. So we've had sulfa drugs for a long time. what they do is target folic acid biosynthesis. So I'm going to show you another graphic of this in a minute, but this is kind of the steps that a bacteria needs to go through to make folic acid. And folic acid is necessary, I got this on the next slide, in order for our cells to make DNA and RNA, as well as some amino acids. Like I mentioned at the beginning of the lecture, folic acid is actually a coenzyme that's necessary for these enzymes here. There's 3 or 4 of them in the pathway here to work to make, we're just going to call this folic acid. Okay, so again, if that coenzymes, if that folic acid, I said that wrong. It's not these enzymes that are necessary. The folic acid that's produced as part of this pathway works on other enzymes in order to make DNA and RNA. Okay, so that's the importance, that's the so what of whether the cell has folic acid or not. But sulfa drugs actually act here on the first enzyme, and don't memorize the name of that enzyme. You don't need to know the substrates there. Just know that it acts on the very first enzyme in that biosynthetic pathway. And we often, to inhibit, okay, so it actually inhibits the production of folic acid. So here's a structure of a sulfa drug, sulfamethoxazole. It's got that sulfonamide functional group there. That tells us that it's a sulfa drug. There is another drug called trimethoprim that works on another enzyme in that folic acid pathway. So usually we just give a sulfa drug and trimethoprim, we market those together, and that's what XXSXT is, sulfamethoxazole and trimethoprim. All right, so SXT is one of the drugs that you use in the curvebauer test if you're in lab. The trade name for SXT is Bactrim. All right, so probably it's not very commonly prescribed, but if you see Bactrim, that's a sulfa drug. Okay, and again, we usually work them together, use those drugs together because that's going to hopefully delay the bacteria resistance and be more successful. And if we're taking out 2 enzymes in the pathway rather than just 1. All right, so here's this graphic is helpful as well, that your book gives you, 10.6 here. So it's got the structure up here of several different sulfa drugs. You can see they all have the same thing in common, this pinkish shaded region here. That structure looks the same as PABA, para-aminobenzoic acid. Okay, that's the substrate that fits in the first enzyme. So here's the enzyme of the folic acid biosynthesis. Okay, so when the substrate fits into the enzyme, then it's a little bit more complicated than that. There's several steps, but eventually you get folic acid, right? But if one of the sulfa drugs binds there, that's what this is showing here. If one of the sulfa drugs binds there instead of the PABA, right, then you're not going to get folic acid. All right, so this is an example of competitive inhibition. Okay, where the sulfa drug is binding to the active site of the enzyme and keeping the substrate from binding there. All right, so then you don't get the product. And apparently it can bind there for a while and then kind of come loose. So you have to kind of keep the concentration of the sulfa drugs up at, a certain point. Okay, so the so what here, that they draw the pathway out a little bit more simplistically. You have to have folic acid in order for you to the cell to be able to make DNA and RNA. All right, we don't have this pathway. We still need folic acid though. We just get it from the food we eat. Bacteria have to make their own. All right, so these, like I said, these are synthetic drugs that were made before we discovered penicillin. We talked last time, Gerard Domach, he developed them in 1932, systematically, just going through lots and lots of different chemicals, trying to find something that had some activity against Streptococcus, I believe. Very broad spectrum. Why aren't they in too much use anymore? There's pretty high incidence of allergies. I looked that up yesterday, and I know I gave you the number. Oh, I said about 10% of it are allergic to penicillin. Some other sources put that number quite a bit lower, maybe around 2 to 3%. And apparently that number is pretty close to be about the same amount for sulfa drugs. Although I bet a lot of you have never actually been on a sulfa drug to know whether you're allergic to it or not, because we just kind of prescribe other things now. Typically, if you do have a sulfa allergy, it manifests itself a little bit more seriously right from the beginning. So maybe that's one of the reasons they're avoided. There still are some specific uses, urinary tract infections, acne, some protozoan infections, still widely used in vet medicine. Okay, so questions. That's questions, comments. That's kind of a rundown of a lot of different drugs. All right, so going back to that concept map, you know, just go in and kind of fill in those, and I would practice doing that. You know, hide it, and you know, okay, what were the mechanisms that we talked about, and what were some examples of those drugs? Okay. All right, so I would like to talk a little bit now about resistance. Okay, so we've alluded to that a little bit already. Thinking first, big picture, how do we think resistance comes about? And then hopefully try to understand some of the molecular mechanisms that happens. All right, so kind of big picture, how does a strain, how does a population of bacteria become resistant? Usually within any kind of a population of cells, say in a petri dish, most of them are going to be susceptible to a drug. You might have one or two that have had some mutation that actually confers some advantage to them and maybe makes them more likely to survive in the presence of an antibiotic. But oftentimes those mutants may not be as fit, so they're not necessarily going to outcompete all the other cells on the plate. So oftentimes they would just kind of die off. But if the cells are exposed to that antibiotic, okay, we would call that selective pressure. Okay, then all those cells that are normally susceptible to that antibiotic are going to die out, and you're going to just have left the couple that happen to be resistant. Okay, and then those are going to proliferate and probably take over. Okay, so where do we find selective pressure in our society today? Healthcare settings, hospitals. You walk into a hospital and almost every patient is going to be on antibiotics. If they're having surgery, they're going to get them prophylactically and after the surgery as well. So hospital settings, lots of selective pressure. So it's no coincidence that we see, you know, the really nasty antibiotic resistant infections, finding them in hospitals most likely. Okay, we tend to like to, when we name strains of bacteria that are resistant to antibiotics, we tend to kind of give them this alphabet soup, these abbreviations. So a couple that we've talked about: methicillin resistant Staph aureus, MRSA, Vancomycin resistant enterococci, VRE. Tuberculosis, we have a terrible time with resistance. In tuberculosis, there are about 1% of all new TB cases are resistant to every drug that we have. You know, gonorrhea, it's like, it's a huge problem. It was probably you know, ranked right up there to the top of our infectious disease worries before COVID, and now it's kind of not talked about as much anymore. How did we get here when to the point where some of our antibiotics aren't working anymore? Again, a lot of selective pressure. We are using antibiotics a ton. Maybe we're using them too much. That question has certainly been asked. I think that we are getting a lot better about misuse. 20 years ago, they were just, you know, prescribed, okay, whatever. I don't know what it is, take this antibiotic, see if it works. There's a lot less of that going on. In fact, when I took my daughter in for an ear infection this weekend, I thought the doctor was going to say, he started talking about warm compresses and taking this allergy medicine, and I thought he was going to say that's all he was going to do for us, but he said, oh no, you need an antibiotic, and I was kind of relieved. But you know, obviously sometimes doctors might prescribe something just to make their patients happy, or patients are, you know, my kid is miserable, just give them something, you know. So there is that pressure there. Worldwide, certainly antibiotics are not prescription. You can just buy them in any pharmacy. So people don't take the right dose, and they're definitely misused in other countries. I would say probably more so than what they are in the US. And this practice of giving antibiotics to all farm animals is, you know, I don't know. We use it as a preventative to increase profits, but a lot of people think, well, that's how we got here. We don't have the experiments that says, yes, this is the link, but it's a suspected, you know, huge factor in contributing to more resistance. All right, so what can we do about it? Well, number one, if you are prescribed an antibiotic, take the full prescription. I know my dad is really famous for this. He'll have a gum infection and he'll take like 8 days of his antibiotic, and then he'll save a couple days worth for the next time he has an infection. Like, then you might not kill every single thing, and they might come back worse, and that antibiotic might not work the next time. So unless you're having some severe allergic reaction, or any allergic reaction, actually, you know, call your doctor, but take the full prescription. And don't, you know, limit the use. This is a really hard one. If you think you might have strep throat, you don't want to control that with drinking cider vinegar or something. Like, be on an antibiotic to get rid of it. But you know, as much as possible, we do want to try to limit the use of them. We do try to use antibiotics in combination, because sometimes they're going to be more effective when we use them together. So here's an example over here where you've got two different antibiotics, and in the area where the antibiotics are both diffusing out, you can see kind of a larger zone of inhibition there. So sometimes antibiotics will have a synergetic effect. Well, they'll work better when you mix two of them together. That's not always the case. For beta-lactams, you need the bacteria to be actively growing. So you don't want to pair that with an antibiotic that's going to stop the organism from growing, right? So then they might be deleterious rather than synergistic. So there's some examples there where, you know, and obviously if we use drugs that have different mechanisms in combination, we're hoping that slows down resistance. Ideally, we're going to use a drug that's only targeting the thing that we're trying to kill. So there's lots of research there. Drugs by design. So we're trying to design drugs that only work against, say, maybe even one organism. Right? And then we're always, you know, kind of messing with the variable group of an antibiotic to try to give the drug a little bit better properties, or maybe avoid resistance for a little bit longer. Okay, how do bacteria acquire resistance? So how does, you know, where do they get this information? Yeah, they might have a mutation. We think pretty often that might happen by horizontal gene transfer. So remember, 3 kinds of horizontal gene transfer. There's a lot of you were mixed up about this on the last exam. So transformation, that's horizontal gene transfer, where you have a dead or dying cell that releases its DNA. Cell walls break up when the cell's dying. And then some other cell can take that up, can take that DNA up and maybe incorporate it into its own chromosome. Transduction is when you have a bacteriophage transferring DNA from one cell and then infecting another cell. And if it's a lysogenic bacteriophage, then that DNA from one host cell can maybe get incorporated into another host cell. But we think most often this happens through conjugation. Especially common with conjugation. So remember, that's when one cell has that fertility plasmid that enables it to extend a pelus to another cell. They don't really show that very well in this picture, but it will share maybe not only that F plasmid with another cell, but it might share some other plasmids as well, like a resistance plasmid, where it might have the information to make some kind of enzyme that would enable the new cell then to also be resistant to some antibiotics. So we think conjugation is probably the most common way that cells share the ability to become resistant. Okay, so what are a couple examples of mechanisms that a cell actually becomes resistant? So we're going to talk about really only 3 mechanisms. And I know that it gets a little bit complicated, like who's doing what. Remember, you have the bacterial cell, you have the drug, and then there might be, some kind of other enzyme involved. Okay, so how do bacteria basically survive being in the presence of an antibiotic? Mechanism 1 is sort of common sense. We're going to try to limit the exposure or limit the access of the drug to the bacterial cell. So again, the bacteria is trying to survive in the presence of an antibiotic. If it doesn't let the antibiotic in the cell, then it's going to be more likely to survive. That's kind of common sense. So how can it limit the access of the antibiotic to the cell? It might just have a little point mutation in the porin structure. So remember, gram negatives have an outer membrane, and porins are those transport channels in the outer membrane. Okay, so only gram negatives have these, because only gram negatives have that outer membrane. And if they can alter that porin structure, maybe it's just an amino acid here or there that's different, so that it doesn't allow the antibiotic to penetrate through that membrane. Okay, it's going to be more likely to survive. All right, so decreased membrane permeability. Some organisms actually have a way to pump out the antibiotic if it does gain access. And these kind of mechanisms are in place already. Some organisms, if they're exposed to certain toxins, can pump it right back out again. And that takes ATP to do that. But they can have the ability sometimes to do that. Pseudomonas is really good at that. If you're in lab, you saw that Pseudomonas was resistant to a whole bunch of antibiotics. It's really good at just kind of limiting the access and pumps it right back out again. And we think that this is a common way that organisms can become multi-drug resistant. Okay, pump lots of different antibiotics out. Okay, another way, I know we're getting close on time here, enzymatic inactivation of the drug. So we talked about this one already. Some cells acquire the ability to make beta-lactamase. And there's actually lots of different variations of beta-lactamase, but remember that cuts the beta-lactam ring and opens it up. And so now that drug doesn't bind to transpeptidases, and it's not going to have any effect on the cell wall integrity. There are other enzymes that can modify drugs as well. I did a little search of the literature. Here's an example of an aminoglycoside, gentamicin, where organisms have been found to be able to change the functional groups of the drug. So functional groups, things like, here's an amino group. There's some other ones here, hydroxide here. They found instances where the organism could change those functional groups cleave them off and maybe, you know, add a phosphate there instead or something like that. And then the drug is not able to bind to the ribosome like it normally would. So basically, another way that the organism can inactivate the drug. Okay. All right. So next time we'll start with the last mechanism where they can modify their own structure. I probably won't post any videos. I'm pretty happy with as far as I got today. Do something fun over your break, and I'll see you a week from today. Work on your homework too, though.